Chapter 4, Staining. The learning objectives for this topic are: explain the purposes of a smear, heat fixation, and chemical fixation in the preparation of a specimen for microscopic viewing. Describe the uses of acidic and basic dyes, mentioning ionic bonding and pH. Describe the simple, gram, acid-fast, and endospore staining procedures. Explain how stains used for electron microscopy differ from those used for light microscopy. So in this topic we're going to cover preparing specimens for staining, principles of staining, simple stains, differential stains, and special stains. When we're going to prepare a specimen for staining, for viewing with a light microscope, there are certain things that we have to do, and you will be doing these in lab. The first thing that you do is you make a smear. If you are pulling a sample off of a solid media, we add a drop of water, take a little bit of material from the solid media, mix it in. If you're pulling a sample from a broth, you don't need to add liquid because the broth is already liquid. So you make that, you spread it out, hopefully getting part of the sample that is 1 cell thick. Then you let it dry. You always air dry. Never use a Bunsen burner to help dry. One, if you do, you're going to get the liquid to boil. That's going to cause bits of live bacteria to go into the air. We call that aerosolization. You really don't want to be breathing that in. Also, if there's boiling fluid, that's going to cause artifacts to show up. And we want our bacteria to stay as much the same as we possibly can get. Then we fix the sample. In the lab, the most common method of fixing or sticking the smear to the slide is by using heat. We call this heat fixation. You can use chemicals to stick the sample to the slide. You won't be doing that in this lab. Here are some basic principles of staining that you want to keep in mind as we go through this section. Cells in cytoplasm are generally clear. They lack contrast. As you remember from the microscope topic, they're really hard to see on a bright field. We add stains. Now stains are generally a salt. That's a basic portion, an acidic portion that have been put together. You make water, you end up with a salt. So stains are the salt with the colored portion, the chromophore. Now most stains are basic, meaning the chromophore picks up a hydrogen ion, which means they have a positive charge. And most cells are, that's right, they have a negative charge, so the basic chromophore is attracted. So most stains that we use, the basic stains, stick to the actual cell. Let's start out with simple stains. Just as a reminder of what something would look under a bright field microscope without staining, this is what it looks like after we've applied a stain. Now we've got contrast. It's a whole lot easier to pick out the cells on the stained version. Simple stains, you just put one basic stain on, and the purpose is to see cell shape. See, we have some bacilli, we've got some cocci, and arrangement. Here we've got some streptobacilli, we've got some staphylococci. A whole lot easier to see than the unstained sample. Now on to differential stains. Differential stains use more than one stain, and they tell you something about the specimen other than cell shape and cell arrangement. So as a reminder, there's our simple stain, and let's go with the most famous of differential stains, the gram stain. Now see, here we've got two different colors. Here we've got the same color, different shades or different intensity, but here we've got red and we've got blue. Something has happened that some of the cells pick up the blue stain and some pick up the pink stain. Now we went over this in chapter 3 as to why some retain the purple stain, the purple primary stain, and are gram positive, and why are stained by the secondary or red stain and are gram negative. So let's take a look at the 3 types of differential stains that you're going to be doing in lab. The first, the gram stain. You're going to get really good at the gram stain. Next we have the acid-fast stain, which we use 2 stains to determine which cells have mycolic acid in their cell walls and which cells do not. So those that have mycolic acid in their cell walls are acid-fast positive, and confusingly enough, we use a red stain for that part, for the primary stain. And those that lose the primary stain do not have mycolic acid in their cell walls, and they're acid-fast negative. Then we have endospore staining. Here we've used two stains. One stain to stain endospores, which are very hard to stain. If you remember from chapter 3, they're wrapped in multiple waterproof layers. Very hard to get a basic stain to stick to that because the endospore has a neutral charge. So we use a special staining procedure to stain the endospores. Then we rinse them. Once you've gotten the stain into the endospore, it's not coming out. But it comes out of the mother cells and the vegetative cells. So then we use a secondary stain, the red stain, to stain the parts of the cell that are not endospores. Now we do have some stains that could be considered differential. Sometimes we're using two stains, sometimes we're not. But let's take a look at these special stains. First is the negative stain, of which we have an example of the capsule stain. A negative stain is where the chromophore is acidic rather than basic. It gives up a hydrogen ion, so it has a negative charge. Well, the cell has a negative charge, and so the stain is repelled from the surface of the stain, and it colors the background. Now in the capsule stain, we add a basic stain afterwards to stain the cell inside, and this causes the capsule to show up. The reason why this works is capsules don't pick up stains very well. We stain the cell, which does pick up a good stain. We stain the background, it leaves the capsule, which is hard to stain, as a clear area. Now when we do the capsule stain in lab, make sure you keep an eye on this kind of thing. This cell is not positive for a capsule, but we still see a clear area. During the process of staining, oftentimes the cell will shrink away from the negative stain. The stain's being repelled, the cell shrinks, and so you get a little bit of a clear spot. That little bit of a clear spot is not a capsule. It's kind of hard to keep an eye on that, but do keep it in mind. Flagellar. We have stains that coat the flagella of bacteria. It's kind of like putting mascara on. Just like mascara makes your eyelashes thicker and easier to see, coating the flagella in a stain makes them thicker and easier to see. This is important when you are using the presence and placement of flagella to identify bacterial species in the genus. For electron microscopes, you have to use heavy metals, generally speaking. It makes sense. When the electrons are deflected and we get dark, dense areas, that's from electron dense areas. We stain samples that are intended for viewing under the electron microscope with heavy metals. Well, that's it for this topic. So here's a reminder of the learning objectives for this topic, and we'll meet up again for the last one, and then we'll be done with this chapter.